morning, good afternoon, and good evening to all our speakers, chairs, and the wonderful audiences in different parts of the world. Welcome back to yet another session of very educational lectures for you. The speaker for the first session of today is a distinguished and honored guest who is a senior faculty from India, Professor Dilip Panikkar. Professor Panikkar is one of the most famous faces of neurosurgery in India, who currently serves as the professor and head of department of neurosurgery at the Aster MedCity Hospital, Cochin, India. He had a long, outstanding career earlier, having served in prestigious institutes like Sri Chitra Hospital, Trivandrum, as well as Amrita Institute of Medical Sciences, Kochi. He is an important member of the Neurological Society of India, as well as the Skull-Based Surgery Society and Stereotactic and Functional Neurosurgery Society of India. We are extremely honored to have him today at our webinars, and today he'll be talking about surgery of the tumors of the supplementary motor area. The speaker for the second session of today's webinar is our honored guest from China, Professor Hu Yang. Professor Yang is an investigator in the Department of Neurosurgery who was in Hospital Fudan University, Shanghai. He received his PhD from University College London in 2013 and his postdoc training at the UT Southwestern Medical Center. Dr. Yang's work revealed the molecular mechanisms of IDH mutation in gliomagenesis, a cover story in cancer cell. His research focuses on the regulation and the role of metabolic homeostasis in cellular transformation and tissue damage, with the goal of using this knowledge to develop novel treatment strategies for brain cancers. We are extremely honored to have him today at our webinars, and today he'll be talking about metabolites as signal molecules in glioma. The chair for the first session of today's webinar is our honored guest from the Czech Republic, Professor Vladimir Benes. Professor Benes has held the position of head of neurosurgery in the Central Military Hospital, Prague, and the chairman of Department of Neurosurgery in the First Medical Faculty, Charles University, and he was a past vice president of the European Association of Neurological Societies till 2007 and the chairman of the training committee of the ACNS as well as the president of the ACNS. He was the previous head of the neuroanatomy committee of WFNS along with Professor Imad Khannan. He is an invited faculty to various workshops and conferences organized all around the world, and his literary contributions sums up in scores of publications in various international journals and book chapters. We are extremely thankful to him for accepting our invitation to chair the session of Professor Dilip Panikkar today. The chair for the second session of today's webinar is our honored guest from USA, Dr. Angela Richardson. Dr. Richardson is an assistant professor in neurological surgery, Department of Neurological Surgery's team of multidisciplinary neuro-oncologic specialists at the Indianapolis. She is enrolled to create an independent research laboratory focusing on gene therapy and oncolytic virotherapy strategies for the treatment of adult and pediatric brain tumors. She completed a cerebrovascular and skull-based fellowship at the University of Wisconsin-Madison, which followed her neurosurgery residency training and surgical neuro-oncology fellowship at the University of Miami. Dr. Richardson earned her PhD in molecular cell biology and genetics from Drexel University College of Medicine in Philadelphia and her undergraduate degree from Columbia University. We are extremely thankful to her for accepting our invitation to chair the session of Dr. Yang. On behalf of the Education Committee of the ACNS and the President, Professor Yoko Kaito, I would like to welcome all the chair speakers and the wonderful audiences to this online platform of ACNS webinars. A very warm welcome to our colleagues in China, and we are extremely thankful to Professor Shubin for broadcasting this webinar on the WeChat channel. Dr. Libun Singh from Malaysia is my co-host for today. With that introduction, I would like to hand over this online podium to our first chair, Professor Vladimir Benes.
Hello, everyone. It's a great pleasure for me to introduce Professor Panikkar, since I've been in Kochi a while ago, and that's really a beautiful place in Kerala, and exceedingly and surprisingly very good neurosurgery there, what I've seen. It's really something extraordinary in India. Professor Panikkar himself is the head of Department of Neurosurgery in Amrita, do I say it properly, hospital. He's associate professor in Sri Chitra Tirunal, difficult for me to pronounce, hospital, and he's senior neurosurgeon in Aster MedCity, Kochi. When I've checked through his PubMed hits, which are numerous, I have found that Professor Panikkar has a really very wide range of interest in neurosurgery, starting with functional, going through pediatric, down to skull-based and vascular, endoscopic, endonasal, whatever you name it. But still, probably the most prominent there were the gliomas. And today he is going to talk about the topic which is really interesting to any neurosurgeon, since we all know the consequences of surgeries in the supplementary motor area. We all know that the outcomes are variable, that the patients may get hemiplegic and then they improve over time, that it's not a direct damage to the motor gyrus, but just to the supplementary area. And I really am looking forward to hear the lecture about these tumors. Professor Panikar, please.
Thank you, Dr. Benis. Thank you, Raja, for inviting me to speak about on this topic. So as Dr. Benis has said, the supplementary motor area is a distinct area that is functionally and electrophysiologically distinct in the brain, has got very unique functions, and for the surgeon who operates tumors in this area can sometimes be the source of great heartburn and anxiety when certain unwanted effects occur. So, okay, let me start off by showing you a case. This is a young lady with a tumor that most of you would have seen or encountered at some point in your career. This is glioma and located right in front of the primary motor cortex. This lady has presented with seizures only, and as is a standard practice, she underwent tumor surgery with an awake craniotomy with motor mapping. But during the course of resection, we noticed a sudden drop in the right lower limb power to grade 0. And this was of great concern. But what was reassuring was that subcortical stimulation at the posterior margin of the resection cavity continued to yield lower limb movements at a very low stimulation threshold, and the tone in the lower limb continued to remain normal. This was reassuring for us, and leaving behind a thin rim of tumor in this area, the surgery was terminated. Very soon after the surgery, on day 4, she started getting better, and within a couple of months, she made a near complete recovery. And this is a post-op scan showing a thin rim of tumor at the posterior edge of the resection cavity. So, as far back as in the 1950s, Penfield and Welch described this area, as they said, just in front of the motor area of the cerebral cortex of man, there is a supplementary representation of movements. This supplementary area is rather well circumscribed and is situated largely within the longitudinal interhemispheric fissure. This was as long back as in the 1950s, in the early 1950s. And in 1977, Laplane gave us a first description, the first clinical description of what we now know as the SMA syndrome, where he described in a series of patients 3 stages of sequential events, which were characterized by an immediate postoperative contralateral hemiplegia, a rapid motor function improvement at variable time frame after the event, and in some of the cases, a mild long-term sequelae involving dysfunction of alternative hand movements, but the majority of the function having returned. So he named this as the SMA syndrome, and this was peculiar to lesions that affected the supplementary motor area. Now, what is a SMA? What is a supplementary motor area as we know it? This is an anatomically defined area of the brain located just in front of the primary motor cortex. The posterior edge of this, being the precentral sulcus, can extend as far as 5 centimeters anteriorly to the precentral sulcus. And it extends onto the medial edge of the hemisphere and comes down up to the cingulate sulcus. Laterally, it extends up to the superior frontal sulcus. This area is further divided functionally into a pre-SMA and an SMA proper by an arbitrary line, what is called the VCA line. It's a perpendicular line drawn from the ACPC line at the level of the anterior commissure. So the pre-SMA line just anterior to the SMA proper. What are the differences? Even though these two areas are anatomically adjacent to each other, they are functionally and in terms of connectivity quite different from each other. The SMA proper having more connections to the primary motor cortex, to the posterior cingulate, superior parietal lobule, and part of the insula, and projects directly and has direct projections to the spinal cord. Receives input from the motor domains of the basal ganglia and cerebellum, and shows a somatotopy with the face anterior, upper limb, and lower limb coming posteriorly. On the other hand, the pre-SMA has greater connections with the prefrontal cortex. It does not project to the spinal cord, receives input from the non-motor domains of the basal ganglia and cerebellum, and lacks somatotopy. And this is more connected with cognition rather than motor function. Again, showing the connectivity, the thalamic input, the thalamic connections to this area are well shown, and the reciprocal connections with the striatum. Looking at the white matter connections, again, this area is very richly connected. The short association fibers, the long association fibers with the cingulum, the superior longitudinal fasciculus 1, the SLF1, and the unique tract, what is called the FAT, the frontal ascending tract, about which we will speak in a few minutes. The projection fibers include the fibers to the claustrum, the corticospinal tract, and the striatum as the frontostriatal tract. And there are commissural fibers that connect both the SMAs across the corpus callosum with each other. The ascending tract, the frontal ascending tract, is a unique tract that connects the SMA with the inferior frontal gyrus. It's present bilaterally. The dominant side has great influence on the verbal fluency in speech, and injuries can lead to difficulty in speech initiation and a decreased spontaneity and fluency, but these usually do recover. The frontostriatal tract connects the SMA with the caudate and the putamen, which is also called the subcallosal tract. It is deeply concerned with the initiation, sequencing, and modulation of voluntary movements, and in the dominant FST also is involved with verbal fluency. This table summarizes all the connections of this area in the dominant and in the non-dominant hemisphere, and the effects of disconnection or damage to these areas. So what are the functions of these areas? The pre-SMA is largely connected with, as these cartoons show, with planning, preparation, selection, and the sequencing of movements. Whereas the SMA proper is more connected with the initiation and the coordination of movements. So as we can see, the initial phases of movements are largely the pre-SMA. The SMA proper helps with the initiation and coordination. These areas are also inhibitory in certain ways, that they can inhibit certain functions. They help in performing memorized motor acts and help in actions like grasping. So the pre-SMA is involved with cognitive motor control, decision making, and it's also involved in cognitively demanding tasks in maintaining attention. Like I said, the inhibitory action of this area is important when in task switching or in stopping a task in motion when it is activated. There are other functions of the SMA which have been described, but in terms of motor control, these are the most important functions of these areas. The motor network, as we know it and has been described, consists of the pre-SMA, which involves, like I said, involves motor planning, the SMA proper, which helps in the initiation and coordination of movement. The primary motor cortex is what issues a final movement command, and the primary sensory cortex allows adaptation through sensory feedback to these areas. The deficits that result from resection or injury to the supplementary motor area can range from either none to involvement of motor and speech. And cognitively, almost all reports indicate there is a significant impairment of spatial working memory. And many of these deficits, especially motor, are shown to be consistent with the somatotopy that is described in the SMA proper. The motor involvement in the motor deficits that results from SMA lesion are very similar to corticospinal tract lesions, except for the fact that tone and reflexes are preserved. The weakness that develops from SMA lesions is often a delayed weakness, which is reported to be as late as 30 to 90 minutes, according to different authors. It also leads to other issues like delay in movement initiation, hesitancy, slowness of movements, discoordination, and a reduction in spontaneous movements. However, the initial report that was given by Laplane has also described the fact that a strong verbal command can sometimes override the deficit, and the patient can have movement when a spontaneous movement is lacking. What is also known and is often exploited clinically is the fact that the dual task response is often affected. And some patients do report a transient grasping reflex or an alien hand syndrome in this. When would you expect to encounter the SMA syndrome during surgery of lesions in this area? It is known that it is reported that the incidence of the SMA syndrome is increased as the extent of resection of the functionally active SMA is increased. The proximity of the resection area to the precentral sulcus, the proximity to the cingulate sulcus, a greater anterior-posterior extent of resection within the SMA. Now, all these are reports by different authors who have attempted to correlate the incidence of the syndrome with the extent of resection. Resections of greater than 90% of the SMA also reported to a higher extent in low-grade gliomas. And when the resection extends to the medial wall of the SMA proper, as compared with resection of only the lateral surface of the SMA proper, the incidence of the SMA syndrome also rises. What is interesting and reassuring is that more than 90% of patients recover from this syndrome over the course of weeks to months. Most of them do recover near completely or completely, but a few may have persistent deficits and paresis, which may be due to either damage to the adjacent areas like the cingulum, the callosum, or tumors that extend to the and involve the primary motor cortex. The mechanism of recovery from SMA injury, from SMA resection, has been variously described as being due to activation of the contralateral supplementary motor area, functional recruitment of the healthy SMA and the premotor cortex, recruitment of the uncrossed corticospinal tract and the primary motor cortex of the healthy hemisphere. What has also been described is the role of plasticity, especially in low-grade lesions or slowly growing lesions, which can lead to a reorganization within this area and limit the consequences of resection from what would otherwise have been a functionally intact area. However, since this mechanism is not does not cannot be used to cannot be ascribed to completely as being the entire reason for recovery, since a recovery from this can also happen in other conditions like vascular insults. Lesions involving the SMA, like this has been described, where there is a high propensity for neoplasms to affect this area. 10% of de novo glioblastomas and 27% of grade 2 gliomas, supratentorial gliomas, occur in this region or involve this region. And there's also a frequent target for drug-resistant frontal lobe epilepsy. The workup and the approach to resection of the lesions involves detailed preoperative counseling, where once the lesion is recognized as having involved the SMA, the patient is counseled, has to be counseled about the high probability that there may be a deficit, but also the fact that this deficit is very, very likely to recover and should not be a major cause for concern. The debate between awake and asleep surgery for hemispheric gliomas continues, but we have at our center, we have switched to almost entirely to awake surgery for most of the supratentorial gliomas, and that has been our preference for many years. If the patient is amenable to an awake procedure. The awake surgery also allows, apart from making motor mapping much easier, also allows a detailed language and other cognitive mapping, which is not possible with the patient asleep. So this is our approach to lesions involving this area. Once a craniotomy is done, all patients undergo a central sulcus mapping using a phase reversal technique to identify where the central sulcus is. And once this is done, the primary motor cortex is identified by eliciting stimulation-evoked responses, the positive stimulation for the primary motor cortex. Further anterior to this, we hunt for the SMA using negative motor mapping in terms of movement arrest and using dual task mechanism, the dual task protocols. The language mapping which we usually do for these include counting and object naming, story narration, repetition, reading in primary and secondary languages. The dual tasks that we use include not only during the initial mapping, but are used continuously during tumor resection. These include a combination of activities like object naming and limb movements, and story narration and limb movements, for example. The fine motor tasks that the patients are often given during this include spiral drawing, copying gestures, fine movements, like, for example, turning the pages of a book, alternative hand movements, and subcortical mapping is used continuously as the tumor resection progresses. This is a standard protocol that we use for all tumors. And these are some examples of dual task, some videos showing dual tasks.
Okay.
Where the patient is asked to narrate a story and at the same time continuously move the hand, and the psychologist is observing this and reporting any changes while the stimulation is in progress. So these are the use of alternating hand movements, where again, these are all sequential fine movements, which the effect of stimulation of cortical and subcortical stimulation can be assessed on these. So as you can see here, there is a difficulty there's a difficulty in performing these movements when certain areas are stimulated. The important thing to note is as long as the primary motor cortex is stimulable with positive mapping, an arrest of movement or alteration of movement with a negative mapping continues to predict an SMA involvement, and a resection of a lesion in this area can continue as long as the positive mapping is elicitable without necessarily the procedure having to terminate as soon as a deficit is obtained. In our brief experience of over 450 gliomas over the last 8 years, we have had 38 gliomas involving the supplementary motor area. 21 were left hemispheric, 17 were right hemisphere. The vast majority presented with seizures alone. The duration of symptoms before surgery was, in the majority of patients, pretty short. And as has been our protocol in the majority of patients, we treated them with awake surgery. So the tumor extensions are important because these sometimes have a bearing, as I had mentioned earlier, these sometimes have a bearing on the possibility of deficits, with a significant number extending to the cingulum, the corpus callosum, and a smaller number extending to the primary motor cortex. The low-grade and high-grade tumors were equally matched in this small series. There was no particular predominance of either. We obtained a gross or a near total extent of resection confirmed on an immediate post-operative MRI in 24 cases. A subtotal excision where the resection was curtailed due to a deficit was in 14. These were the new onset deficits, which we noticed in 18 of these 38 patients, where 3 were language, 11 were motor, and both language and motor deficits in 4 patients. All these patients, all these 18 patients showed a recovery over time, the majority of them showing a recovery within 6 months, but 3 of them showing a very delayed recovery. One of the patients continues to have some issue with fine motor control, but is otherwise functional and back to work. A few case examples here. This is a young man with a large non-dominant side lesion, which was operated with an awake craniotomy. With direct electrical stimulation, the primary motor cortex was identified at 8 milliamperes posterior to the lesion. But during tumor resection, it was uneventful. But during hemostasis, there was a sudden drop in power on the left side, and he became, and he developed a significant deficit. He had a significant hemiparesis. However, the tone was normal, the reflexes were preserved, and the plantar continued to remain flexor. This was a post-resection scan, and this was very typically an SMA syndrome. And this man, as expected, made a complete recovery within 3 months. Another patient, a young gentleman with this very diffuse lesion, underwent awake surgery. Again, the motor cortex was mapped on the surface. Subcortical mapping on the corticospinal tract was stimulated at 10 milliamperes. He developed a right lower limb weakness during surgery, and in the immediate post-op period was noted to have a right hemiparesis with significant right hemiparesis of both the upper and the lower limbs, which the post-op scan showed remaining tumor, but again started improving within the first week and complete recovery within 2 months. Yet another case, a similar lesion excised, stimulable motor cortex, deficit appearing, but improving, and asymptomatic within 3 months. So in conclusion, we should recognize all surgeons who manage tumors, manage hemispheric tumors, hemispheric gliomas, should recognize the uniqueness of lesions involving the supplementary motor area and its unique physiological characteristics. And even though there is a high propensity to develop deficits from resection of the SMA, it is reassuring that the vast majority of these improve and the patients make an excellent functional recovery. What is important is to ensure that the primary motor cortex and the corticospinal tract remains stimulable all through the procedure, in spite of your patient having a deficit. Thank you.
Thank you very much, Professor Benes.
Thank you, Professor Panikar. It was really a great lecture, very, very comprehensive. And I believe that there will be lots of questions. I just wonder, during the awake surgery, you see that something's happening.
Yeah.
But as long as you have no more stimulation, you just go ahead. We never use awake surgeries in these cases because we know exactly this. If you have a motor response, well, you can go down to 0.5 milliamperes. You just go ahead with surgery, expecting some problems after that. So awake just tells you that something's happening, nothing more.
Exactly. I think what is most important here is the stimulation response of the primary motor cortex. As long as you can continue to elicit that at a reasonably low threshold, you can be almost certain that this patient, whatever deficits happen, will almost certainly recover. The problem is in the initial stages, for many surgeons, the moment you encounter a deficit or the beginnings of a deficit, there is this tendency to panic and say, no, I've created a deficit and I'm going to stop the surgery. What you'd often end up is leaving behind a substantial amount of residual tumor, which oncologically is not going to help the patient one single bit. So
Right.
what matters is we try to push the extent of resection, and this is the only way you can do this.
Yeah, yeah. So would you say that the awake surgery is something which might really cause suboptimal resections?
I think it's, see, for the motor, it's probably less important. For the language, certainly it is. But I think depends on what you're comfortable with. We have almost entirely switched to awake surgeries for whether it is for motor or language or other cognitive assessments. The reason for us switching over to awake surgery is the fact that once you do this repeatedly, the entire team, the anesthesiologists, the theater techs, the theater scrub nurses, and everybody in the theater is very clued into an awake surgery, and we don't have any issues with that. So it's a very smooth flow for the patient all through, rather than doing it on very select cases where the entire team can be a little wary. And that is when you're likely to have more of issues with managing an awake surgery.
I see what you mean. I see what you mean. Okay, any other questions from the floor?
Ben from Hong Kong has joined us. Any questions from you?
Yes, and thank you, Professor Panikar, for your great presentation on the SMA. May I ask for, in your experience on those awake surgery, for those having the language deficit, are they exclusively the left side lesion, or you have some experience with the right side? And also, do you have experience in for those who have a multilingual patients? How do you map them, and is there any particular things to concern when you concerning the SMA region? So, and my third question is about the line bisection test. Do you find this task is useful in SMA syndrome or not? Thank you so much.
Thank you. I'll take the questions in sequence. The dual task is something that is strongly recommended for SMA lesions and for surgery in the region of the SMA. And yes, it does give you a lot of value for this. You had wanted to know about. I'm sorry. Your the next question was
about for those having the language deficit. Are they exclusively the left
right,
side?
okay. So we have encountered language deficits on both hemispheres, though the propensity for the dominant side Aslan tract to lead to more of a dysphasia and slowness of speech is much higher on the dominant side. On the non-dominant side, we do encounter a certain amount of loss of fluency, but they can still initiate speech. It's not as bad as on the dominant side. So there is a loss of fluency. There is an interruption of fluency, but on the dominant side, yes, the spontaneity is certainly lost. So we see this during surgery often when you have a patient who's talking very fluently, and you can hear the patient in conversation with the psychologist. A fluent conversation after some time becomes largely one-sided, where it's a psychologist asking a question and the patient only answering a question rather than the spontaneity of conversation that happens. So this is a change that we often notice. And your third question was about the bilingual, was about the multilingual patients. Yes, multilinguals are important. We do have the kind of the population of patients that we deal with are sometimes multilingual in various ways. But what we often look at is this: when the psychologist does a pre-surgical assessment, we look at the dominant language. We look at in what language the patient has been schooled and is most comfortable. And we preferentially use that language for the intraoperative assessment. Rather than as of now, we do not have multiple tasks for people who are very fluent in multiple languages. We just use the language in which the patient is most fluent and most comfortable.
Thank you so much. And thank you for
There is a question on the chat, Professor Panikkar. If apart from gliomas, other lesions in this area that lead to the SMA syndrome?
Yes, it is described with vascular insults, with strokes. It is described an SMA syndrome.
That's great. Yes, my friend Dr. Harshal has joined us today from Mumbai. Yes, Harshal, you can ask.
Good evening. Fantastic lecture by sir. I just wanted to know if there are any specific rehabilitation programs specifically directed to these patients who develop SMA syndrome. And do you have any experience with transcranial magnetic stimulation?
I do not have experience with transcranial magnetic stimulation, but yes, one of the ways that has been described for managing these people is to initiate them on a program of speech therapy and language rehabilitation and motor rehabilitation, even before the surgery starts, even during the phase of pre-surgical assessment. So this is supposed to make the recovery a lot better. Again, it's something that is very, very difficult to practice because most of these patients get into theater very fast once they are diagnosed, and we don't wait too long on them before doing that. The use of TMS is described not only for facilitating rehab, but also for pre-surgical localization of functional areas, which can be linked to the navigation and to use. But we don't have access to the TMS for this, so no experience.
Thank you very much. Yes, my co-host Liu Boon Singh from Malaysia. Liu, can you hear us? Fortunately, he's gone out. Dr. Richardson is a neuro-oncology fellow. We can invite her comments as well.
I thought that was a great lecture, especially the history. Thank you so much. The question I have is more, once someone develops, if they have a deficit post-operatively, do you have a feel for ways to understand how long that's going to last when you're counseling families or the patient in terms of their recovery?
It's a bit of blind prediction that we have to do, but what we try to tell them is to be prepared for weeks to a month or two of deficit. See, the thing is, it's very concerning in the initial days when the patient is still hemiplegic or the movement is zero. There may be a little bit of movement immediately after the surgery, but by day one, it sometimes drops further with some edema settling in. So that is a moment where there's great concern. The moment your patient starts getting a little bit of finger or toe movement, it's reassuring because you know that some activity has restarted. And then the family is reassured that, okay, there is visible movement. We can work on that and get it better. But once they do that, they tend to improve quite fast. So so what we generally do is to tell families to expect a few weeks to maybe a month or so of a deficit, but will depend upon when the initial recovery of movement starts happening.
Professor Panikkar, I have a question. You have shown all the connections of the supplemental area. You have shown the functions and everything. We as neurosurgeons usually are in gross neurological findings, isn't it? But the hand is moving, the leg is walking, whatever you say, hemiplegia, non-hemiplegia. Should we do in these patients really thorough neuropsychological investigation? Would you expect some permanent deficit?
Most of these patients, we do a detailed neuropsychological battery through in the pre-surgical, as part of the pre-surgical assessment. So what we find is the majority of them have a significant executive function deficit. They do have, it may not come to the surface, it may not be apparent, but when you do the standard battery, when you put them through the battery, most of them do have an executive function affection. And the ones that are higher grade will almost certainly have multidimensional involvement in terms of not only executive function, but even visuospatial. That's a correlation that we have found with our cases, but really cannot say. But there is an element of disruption of cognitive function, which you can elicit with a detailed testing.
One question that I would like to put across to Dr. Panikkar is that in the post-operative setting, when the patient worsens, we always have this feeling that whether edema is settling in. Does it prompt you to use steroids? Because I once famously, I heard Professor Benes quoted that steroids is for the doctor, not the patient. So how is it with you?
See, if you understand the physiology of edema, edema is always self-limiting. The post-surgical edema, it happens, it will happen. It happens, it's very predictable, and it goes away. The issue comes when you probably have super added problems like vascular insult. You might have coagulated one artery more than what is necessary. You might have devascularized some area. And these are things that will extend the area of deficit beyond what you would expect from a peri-cavity edema happening. So steroids, again, unless there is a definite mass effect, like we use steroids, but perioperatively, when there is a deficit, but there is stimulation that is there, we don't necessarily extend the duration of steroids. It's still a standard perioperative course. We often use a hypertonic saline infusion just to keep the sodium up a little bit in the perioperative period, and that's it. And then focus more on getting the patient into rehab as soon as possible.
Thank you very much. We have Dr. Liu Boon Singh coming back. Liu, I hope your mic is right.
Yeah. Thanks, Raja. Thanks, Professor, for very nice presentation. Professor, I just wanted to find out from you, how would you handle a patient that when you test motor power absence, but you think that the patient still need further cytoreduction? How you convince the patient that no, I need to go on with my surgery? I just want to know how you tackle this condition. My second, Professor, I show you show that on stimulation there are 8 milliampere and 10 milliampere stimulation for the motor area. Do you use a monopolar or bipolar, and does it really help us to determine the distance of the corticospinal tract from your site of stimulation? Thank you, Professor.
Yeah, we use monopolar stimulation for motor function, and we switch to bipolar when we use language mapping. And yes, there is a distance correlation there. It's described as 1 milliampere for 1 millimeter of distance, but whatever it is, the higher the threshold, the greater is the distance that your functional tracts are away from your point of stimulation. So that gives you this margin of buffer, this margin of safety. If you are very sure of your electrophysiology and your mapping, you can go down. I mean, I have seen Professor Mitchberger using a stimulator in one hand and an ultrasonic aspirator in the other hand, and he keeps alternating between the two, and can get it down to as little as a couple of millimeters. But that depends on how confident the surgeon is to do that. With regard to your first question was about the cytoreduction when you have a deficit.
Yes.
See, that is a debatable point there. You would, as a surgeon, when you're taking out the tumor, you do want to go ahead and resect the maximum amount of tumor. But I think what matters here is protecting your corticospinal tract. If your primary CST is stimulatable. Now, we use the CST stimulation in two ways. One is the moment you identify the corticospinal tract, you can put a strip on the surface and use the electrodes on the strip to keep stimulating the cortex, the primary motor cortex. And then as you go in with the tumor, you're also combining it with the subcortical stimulation with a monopolar. So a combination of these two, as long as you are able to elicit a corticospinal tract function at a threshold that does not change significantly from what you had initially got pre-resection, you can continue to do the resection. So, right. But this is, yes, the surgeons probably gets a major adrenaline rush or during this time, and you have to be very, very confident to continue resection when there is a deficit that is appearing there.
How about facing the patient in awake, Professor? Awake means when patient feel that possibility when you stimulate an area that arrest the motor function, the patient will expect you to stop the surgery. And how do you face in this kind of condition, Professor?
We have a detailed counseling for all our patients where they are taken through all possible scenarios and told about what can possibly happen. What also helps, which sometimes works, is if you can get your patient to talk to somebody who's undergone an awake surgery. And they, hearing from somebody who has been through this, maybe even has had a deficit that has recovered, it's much more reassuring to them than the surgeon telling them that everything is going to be fine.
Agree, Prof. Thank you.
That is
one more question.
Very interesting. Yes, Professor Dennis.
Did you see any differences between low and high grades?
The high grades have a lot more baseline dysfunction than the low grades. The low grades tend to have compensated more, and the deficits that you have, mostly the cognitive deficits, are sort of subsurface. Very few of them come up to the fore. It's only elicitable on testing. But the high grades will almost always have a much greater degree of dysfunction, much more overt dysfunction. And some of them, and these are the ones that do tend to invade the primary motor cortex, and they can present with a hemiparesis initially.
Okay.
Well, thank you very much. That was indeed a very interesting session, and we learned a lot. Thank you, Professor Benes and Professor Panikkar, for your time and support. We can take the final remarks from Professor Benes before we move on to the second session.
Exactly as you said, Raja, we have learned a lot, and this is probably the topic we all know about. And we all see the patients who are hemiplegic after surgery in this area. And now we've seen that it's far more complex, actually, than the motor function. That this really has to do a lot with cognition. This really has to do a lot with, let's say, movement initiation, coordination, and all these features. So it's always good when one starts thinking about the problem in a more complex way, which probably thanks to the Professor Panikar's lecture, we all shall do in the future. Thank you, Professor Panikar.
Thank you.
Well, thank you very much. Our second speaker, Professor Hu Yang, is ready. Invite our second chair, Professor Angela Richardson, to say a short introduction and invite Professor Hu Yang in turn. Dr. Richardson.
It's my privilege to introduce Professor Hu Yang. As we heard earlier, he is an investigator at Fudan University, and he has an impressive publication record, including papers in some of our top journals, Nature, PNS, Cancer Cell. Today, he's going to be talking to us about metabolites in gliomas. And as we all know, cancer cells, one of their hallmarks is they're rapidly proliferating. And so they have changes to their metabolic profile to allow this. I'm sure we've all heard of the Warburg Effect and things of that sort. So with these activation of oncogenes and downregulation of some tumor suppressor genes, these transcriptional, epigenetic, and cellular signaling pathways can be significantly altered. I'm sure he's going to tell us a lot more about that. And so it is my pleasure to introduce him to talk about metabolites serving as signaling molecules in glioma, a lesson learned from IDH mutation.
Thank you. Thank you very much. Thank you. Thank Dr. Xu Bin for the inviting to me to give this talk. And good morning, good afternoon, and good evening for everyone. And today, my talk, I'm from Fudan University, Huashan Hospital. Today, I would like to present my studies on the metabolic regulation of gliomas with IDH mutation and what we can learn from this hotspot mutations. So today, I will share, I divide my talk with two parts. The first part, I will introduce some of my studies of the mechanism of IDH mutation in the tumorigenesis. And the second part, we will to introduce some recent work for the lesson learned from the mutations. In more than 80 years ago, Otto Warburg observed the tumor cells has altered metabolic regulations. Despite having an increased uptake of glucose, tumor cells always produce much less ATP than expect from completed TCA cycles coupled with oxidative phosphorylation. And instead of accumulation of a significant amount of lactate, enhanced glucose uptake provides some basis for the PET technologies, and which has been widely used clinic for the tumor detections. And by injection patients with radio-labeled glucose analogs, and followed by imaging with PET. However, the underlying mechanism of this effect remains poorly understood. In 2008, and the Cancer Genome Project made a surprising discovery in the form of recurring mutations in the gene encoding IDH in gliomas. And this finding had been, after this finding was published, our group has firstly moved on and found that this mutation targeted the IDH1 or IDH2 where abolishes normal activities which produce FKG. And in 2009, Lansing and their co-workers made a surprising discovery that is in addition to reduce the activity of FKG production, the IDH mutation also get a new activities which is in the production of 2HG, and in 2000. Also in the tumor samples, they also found the metabolite of 2HG is highly accumulated. And in 2010, and in our group, and we also set up the metabolite detection assays. We also found when we overexpress the hotspot mutations IDH1 in glioma cells, we could see a significant accumulation of 2Hg and with a decreased level of RFKG in the cell lines. And later, when we found the metabolite 2Hg was structurally similarly with RFKG, except for the oxidative state at the C2, on which the oxygen in RFKG is replaced by a hydroxy group in 2Hg, suggesting the 2Hg may be an antagonist of RFKG functioning in the cell. So we are trying to dissect the mechanisms of how this mutation will cause gliomagenesis. And at the same time, and at 2009, and several groups have discovered the firstly reported the DNA demethylase is called TET proteins, which requires RFKG and oxygen as a co-substrate to oxidative the hydroxy group at the cytosine and make the 5-hydromethyl cytosines. So as we propose the 2Hg may be a structural similarity to RFKG, which can be an antagonist of RFKG. So we hypothesize this 2Hg produced by IDH mutation will inhibits the TET activities, which leads to the hypermethylation group. So we use the dot plot and also the immunofluorescence to detect the products of TET, which is 5-HMC. As we can see here, when we overexpress the mutated IDH1 in the cells, we could see a decreased level of the 5-HMC's productions by dot plot and also immunofluorescence. So this part lead us to make a summary is that tumor-derived IDH mutations will cause the metabolites like 2Hg accumulation, which will inhibit the RFKG-dependent dioxinase, such as the TET families, which is the DNA demethylase, and cause the epigenetic reprogramming, and also and then cause the in gliomas. And in 2010, in Nosma group also reported from the glioma samples, they found hypermethylated groups from this hypermethylated groups from the patients. So when we did the gene mutation correlation, we found the most of the hypermethylation groups show with the co-recurrence with the IDH mutation. So this relevance is consistent with what we have found before. And in summary, we think the normal cell with the IDH white type produce a normal level of RFKG and keep the activities of both DNA demethylase and histone demethylase produce with the epigenetic regulation and keep the long stem cell differentiation. With IDH mutation, we can see the metabolites of 2Hg is accumulates, and with RFKG, with a decrease of RFKG in the cells, and this all of these changes will inhibit the DNA demethylase, and this demethylation will cause the altered epigenetic regulations. So this part, and we have linked the IDH mutation and the metabolites from IDH mutation with as a singular molecule to like inhibit the DNA demethylase or histone demethylase and to induce the tumorigenesis. So at the second part, we would like to introduce the lessons what we can learn from the IDH mutations in the study of gliomas. And so I will ask whether we can see some other metabolites could also bridge the metabolism and metabolism to the tumorigenesis. So, and to answer these questions, and we have to like share with you guys with a paper published in 2009 in Science. Is from Craig Thompson's group. They first point out the metabolized acetyl-CoA, which can regulate the gene expression through the way to acetylate the histones. This work first linked the cellular metabolism to gene expressions. So besides IDH mutation and also paper self-group reports the genes in the TCA cycle such as SDH and also FDH were also mutated in some kinds of tumors, and they will also cause the accumulation of succinate and fumarate. And later, we also provide evidence that accumulation of succinate and fumarate are also competitive inhibitors of alpha-KG. We are also inhibit the histone and DNA demethylase and cause tumorigenesis. And so in 2017, Mary and Julia group also found the accumulation of GHB and other metabolites caused by the enzyme SADH downregulations. Also, we also inhibit the DNA demethylase and cause the glioma stem cell proliferation and inhibits the glioma stem cell proliferation and self-renew. So, and to summarize this part, I think we have found several kinds of metabolite regulation patterns, such as IDH mutation, SDH mutation, or FH mutation. All these kinds of mutation will cause some of the metabolite accumulation and then inhibit the downstream epigenetic pathways such as TET and histone demethylase KDMs. So we are trying to propose and whether we have there's other metabolic intermediates like metabolic genes downregulation and cause the metabolite accumulations will also act as a singular molecule to inhibit the histone or TET DNA demethylase that will link to tumorigenesis. So, but in order to answer these questions, we firstly use a computative method to search for the metabolites which is structurally similar to alpha-KG. Here are the 10 hot hits, and as shown here, we can see the alpha-KG is here, and which has a positive control, and we also found a fumarate and a succinate and or some other metabolites here. So and then we list the structures of all the metabolites here, and we can as like 2HG with alpha-KG, and all the other metabolites are structurally similar with alpha-KG. Some with with short form of the CE, the carboxy, and some of with the oxidative state is a little bit of difference. So we will ask whether all these metabolites could also be an antagonist of alpha-KG in the cells. And then before doing the experiment, we use the computative method to docking these metabolites and into the histone demethylase and KDM4A. And this is the result from a malate molecule to dock with KDM4A. As shown here, we can see the malate can similarly binds with the activated forms of active site of the KDM4A, which showed an inhibition effect here. And later, we're trying to in order to provide the inhibition effect of this metabolized with histone. So we set up a robust assay to measure the activities of histone demethylase, and we synthesized the peptide H3K36 trimethyl peptide, and we incubated this peptide with alpha Kg and also KDM4A enzymes, and use a mass spec to measure the methylation state at the lysins. So as shown here, we when we use different oh sorry, this is 5. I didn't change it. So we use a different concentration of the alpha Kg and incubate with the enzymes. We can see a trimethylated level of lysine is decreased robust here. So in order to measure what we have found before by computation method, so we incubated the succinate and malate with alpha Kg at different levels. As shown here, when we increase the level of succinate or malate, we can see the accumulation of trimethyl level of lysins in this assays. This indicates inhibition of KDMs by this metabolite like succinate or malate. We also find some negative results like the ornithine when we use the concentration as high as 10 millimole, and we didn't see any inhibition of trimethyl lysins in this buffer. So this metabolite we think is cannot antagonize with alpha Kg to inhibit the histone demethylase. So we have some like summaries for the inactivities of metabolites here. We can see here both of these two metabolites with two positive charges, which is not suitable for the binding with the active sites of histone demethylase, and some of the metabolites with too long carboxyl, or some with large shapes. The shapes is too large to dock with the active sites. So we still working on this part for the in vivo evidence. So to summary here, we hypothesize there are many metabolites which is in some kinds of IDH1 type tumors with some metabolic genes dysregulation with accumulation of some kinds of these metabolites where antagonists with alpha Kg, which will inhibits alpha Kg dependent dioxinase, including the histone and the DNA demethylase. This will cause gene expression changes which links to the tumorigenesis. And today I will stop here. So I would like also thanks Dr. Mao Ying and Xu Bin for the support, and also Dr. Liu Ying from Fudan University for supporting my work here, and also some of the students involved in these works. And that's all. Thank you.
Thank you. That was very great talk and reminded me a lot of my undergraduate biochemistry classes. I was wondering if you could comment on what you think this informs in terms of, I don't know if you have a lot of patients who are interested in ketogenic diet for some of these gliomas, and that would alter the intracellular metabolism, and how you think that might be related to this metabolic reprogramming you're seeing.
Yes, from the patient part, yes, we are trying to find more conclusions which will fit our models. And the IDH mutation, I think, is the most significant one. And for other part, we have some conclusions And some of the patients which originate from like a gene regulation, like as I mentioned, the T-cell cycles which we also found like SDH and also FH mutations, but in some tumors, we don't see the mutations. We do see the down regulations of FDH or FH or SDH, which will also cause the accumulation of some of these metabolites. I think in IDH wild type tumors, some of the patients is like it's not a gene mutation work, but it's the gene expression level will cause this the same phenomenon. So it's better to use the gene expression to find these patients, and maybe the tumorigenesis mechanism is through this like the same paradise of this mechanisms.
Yes, Ben has raised his hand. Ben.
Hello, Professor. Thank you and congratulations on your research work. And my question is for your my first question is about your SH and FH mutation type. Do you have some clinical data about their prognosis? Is that are they carrying a better prognosis like the others mutation? And my second question is there any druggable targets for your based on your research result? Thank you.
Okay, very good question. Thank you. Yeah, the first one for SDH and FH mutations. Yeah, we found it's not in gliomas. It's in other kind of tumors like renal carcinomas, and the mutation rate is very low. And the cases here and all over the world, I think in 2019, it's low more than 1,000 patients all over the world. So it's very rare mutations here. So for this part, we think like some of the tumors with down regulations, the expression of these genes will contribute to these mechanisms. And in the second, your second question is for the druggable for the translational part. Yes, and in the IDH mutations, I think many people know it's also mutation. It's also highly mutated in the AML, and the famous company from Angels in US. They also they disrupt a drug which is specific targeted the IDH mutation, and this drug can they have some like clinical trials on AML patients with significant improve of the patient survival. So this drug has been approved by FDA used in the AML. However, in gliomas, the clinical trial is not that successfully. So it's still going on. And I think there's some heterogenesis of IDH mutation patients. So we need to try to figure out which patients we are suitable for the IDH mutated targeted therapies.
Thank you
very much. Is my co-host Liubun saying any questions from you?
Yeah, I don't have any question, Professor.
Understood.
So
I think there are no further questions. We can get the final remarks from Dr. Richardson.
Oh, well, thank you so much for that talk, really demonstrating the metabolism and the relationship between IDH and these metabolites and tumorigenesis. And I think it really shows the power of some of the computational work you've done, which I think is really fascinating. So thank you so much for sharing with us today.
Very much.
Okay, thank you.
Thank you very much. Yes, Dr. Panikkar and Professor Benes, any comments from you?
There was an exceedingly good seminar, as always, with ACNS. So I just hope that, Raja, you will continue in these endeavors, because they are extremely valuable and extremely helpful. So thank you for organizing this.
Well, thank you very much. I will close this officially now on behalf of the Education Committee of the ACNS and the President Professor Yoko Kato. I would like to thank both the speakers of today, Professor Dilip Panikar and Professor Hu Yang, as well as the chairs, Professor Vladimir Benes and Dr. Angela Richardson, for the time and support for the ACNS webinars. I would like to express my sincere gratitude to Professor Shubin for broadcasting this on the WeChat channel, and also my co-host Dr. Liu Boon Seng and Ben, who is joining from Hong Kong as well. So until we all meet tomorrow online, it is bye-bye from all of us. Thank you very much for joining.